A study of the cytoskeleton malignant cancer cells using GFAP, vimentin, F-actin and microtubules. Cancer is a group of over 200 diseases which affects a huge proportion of the population. As there are so many different cancers, it is very hard to find a drug which could treat all. There are 6 hallmarks of cancers, and treating one of these could mean all cancers could be cured. 2 main factors make it hard to treat cancer. One is that cancerous tumours can metastasize to other areas of the body, and the second is that cancerous cells are our own body cells and so display self-antigens, making them hard to target. Metastasis is one of several hallmarks of cancer. Once the tumour is developed, the cells migrate to the edge of a blood vessel. Then intravasation occurs where they enter the blood vessel. Transport then occurs where it moves through the vascular system. Then extravasation where the cell moves out of the blood vessel. Metastatic colonization then occurs where a new tumour develops. As the cytoskeleton is involved in cell motility, it is very important in many of these stages. I'm going to look at intravasation and identify how it could be targeted. This image shows the positioning of the components of the cytoskeleton. Microfilaments are a component of the cell cortex and so regulate the shape and movement of the cell's surface. Intermediate filaments bear tension, maintaining cell shape and rigidity, and microtubules support the cell and also provide a route for organelles to move throughout the cell. I used immunocytochemistry to qualitatively analyse the localizations of the cytoskeletal components. I tagged the antibodies using immunofluorescence and then used a confocal microscope to gain images. I used indirect immunofluorescence, and so first attached a primary antibody. Onto this I attached a secondary antibody which was conjugated to a fluorescent dye. This tags the antigen fluorescently and so can be viewed under a confocal microscope. To gain quantitative data of the presence of the cytoskeletal components, I used flow cytometry. I used immunofluorescence to tag the specific antigens and then pass the suspension into a flow cytometer. I used the data recorded to find the fluorescence intensity and the mean percentage gated. Fluorescence intensity shows the amount of antigen being expressed, whilst percentage gated shows the number of cells expressing it. But which cytoskeletal component could be targeted in an attempt to treat cancer? I have looked into the localizations of the cytoskeletal components to see if they agree with the ideas set forward on how the cytoskeleton aids intravasation. I then identified the component I believe to be best to target in the hopes to treat cancer.